This is Jimmy's video about fragment-based drug discovery.
La 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 la, wait till I get my money right.
Alright, I suppose I'd better get started on this fragment-based drug discovery project. It is due in about 2 hours after all. What even is a fragment? A fragment is a small molecule, such as a small protein, which has certain desirable properties that might be useful as part of a drug. Whoa! Thanks, Jimmy. But if you have a small molecule, how do you know if it might make a good fragment?
Hey Jimmy, there's a rule of thumb for good fragments called the rule of three, introduced by Giotti and colleagues in 2003. The rule says that fragments should have the following characteristics: molecular weight less than 300, C log P of at most 3, this is a measure of hydrophilicity, and the molecule should have at most 3 each of hydrogen bond donors and acceptors.
Thanks for that explanation, Tina. I think Inez wants to say something.
Jimmy, I have a big question. What is fragment-based drug discovery?
That's a great question. I wonder if Brianna knows.
Hello, Jimmy. Fragment-based drug discovery is a method for finding molecules which would make good drugs. In contrast to other methods, which are usually based around finding a whole molecule and possibly fiddling around with it and usually only making it smaller, this fragment approach starts with small bits and builds it up from there. This also gives the person designing the molecules more room to play around, as they can both add and subtract the molecules they're working on.
Thanks, Jimmy!
Yeah, thanks, Brianna. I mean, me. Look, here comes Danitza.
What are the advantages of fragment-based methods?
Oh God, I wish I knew!
Ciao Jimmy! Le molecole di droga guida trovate usando metodi tradizionali sono spesso grandi e poco solubili. Un metodo basato sui frammenti può evitare questo problema.
Okay. So it can produce molecules which are more practical as drug candidates. But it sounds like a big job.
No, Jimmy. A fragment-based approach makes it possible to explore a greater part of the space of possible molecules in fewer experiments than traditional approaches.
I see. Making it more efficient to explore the space of molecules does sound convenient.
Thanks, Jimmy.
This all sounds good, but my background's in computer science. Is there any room for computer stuff in all this? Oh, cool. So what might these computational methods involve?
Hey Jimmy, once we've identified some fragment with the priority we need, there are two main things we can try to build these fragments into a potential drug molecule, growing and linking. Firstly, we can grow the molecule by adding new bits on until it takes the desired form. Secondly, we can link existing fragments together. If we can fix the right molecule parts to link them with. The difficulty here is that the structure of the molecule makes a crucial difference. Even the angle between the fragments are very important, and it can be very difficult to link them in a stable way with the correct shape.
Thanks, Iyad. George, can you tell me about any particular computational method?
Buongiorno, Jimmy. One computational technique is called fragment replacement and molecular design, and it is implemented in software packages like Broad. It works by replacing fragments or other parts of the molecule with other, slightly different ones, a little bit like a perturbation step that you might see in other algorithmic approaches, like Monte Carlo Markov chains methods.
Oh wow, cool! Uh-oh, here comes Kaiser.
Why do we bother with these computational methods?
Barsor, Kaiser is being a skeptic again. Can you help him out here?
Marhaba, Jimmy. Since computational methods can be used to find and design fragment-based molecules, scientists would waste less time experimenting with molecules that are no good and instead have more confidence that the molecules they do test are very good. Thus, anyone who is doing his PhD in a biochemistry or a related field would take less time. For example, instead of 3 years, it would take only 2 years to finish.
Thanks, Jimmy.
Okay, now I'm sold on fragment-based drug discovery. But say I wanted to build a fragment library. How might I do that?
Jimmy. Fragment-based methods usually involve the use of fragment libraries, basically databases of fragments and their properties. This can speed up the development process, as it's possible to make use of previously developed knowledge, but the process of creating that database in the first place is a lot of work.
Hiya, Jimmy. When building fragment libraries, it's important that we have good knowledge about the structure of the fragments that go into the library. To discover or confirm these, the screening methods that are used include X-ray crystallography, nuclear magnetic resonance spectroscopy, and mass spectroscopy. Kashunga!
Wicked! Thanks everyone!
This is Jimmy's video about fragment-based drug discovery.